All right, well, thanks for joining us today. My name is Jay Scott. I'm one of the co-executive directors at Alex's Lemonade Stand Foundation, and we have a really exciting lecture for you today. Just some ground rules. If you have any questions, please type them in the chat box, and we will have Casey answer them at the end. If you have suggestions for other lecture topics, please put them in the survey that will be posted to the chat. It's really a big help for us when people that are on these talks give us new ideas for other lectures. And then coming up on Thursday, we have a follow-up to this lecture by Dr. Jackie Taroni, who's the lead scientist at the Childhood Cancer Data Lab. And the topic, or the title of her lecture is Unsupervised Transfer Learning for Rare Disease Transcriptomics. But today, we're lucky to have Casey Greene. Casey Greene is an associate professor at the Perelman School of Medicine at the University of Pennsylvania. And he's also the director of the Childhood Cancer Data Lab at Alex's Lemonade Stand Foundation. His research focuses on harnessing large collections of biomedical data to discover new physiologically relevant patterns. He's also the brainchild behind what we do in the direction we go at the Childhood Cancer Data Lab. So with that, I will turn it over to Casey. Welcome, Casey.
Thank you, and thanks for having me. When I was asked to do this, I thought it would be a good opportunity to use the time to give a little bit of an intuitive introduction to essentially machine learning and deep learning and how they're used in biology and medicine. I know many of you and I often get asked to review papers, serve on grant panels, and write pieces and think about what's going to happen in the future and how large-scale data is going to transform both research and practice. And over the last few years, I've sort of been putting my thoughts together, and I thought this would be a nice opportunity to kind of share them at the 101 level, kind of primarily aimed towards biologists and clinicians, to give just a little bit of jargon and familiarity with jargon, and then also to talk more about some of the intuitive issues that I think we need to think about deeply as a field, and that's going to take all of us, not just machine learning experts, not just biologists. It's really going to have to be everyone working together to really make sure these technologies are as beneficial as they can be. So first, I kind of want to talk briefly about sort of types of algorithms. And when we think about machine learning algorithms, essentially what we're doing is we're starting from data and then we're trying to answer a question. And there's really a few different framings of questions, but the two we see most often are essentially those asking kind of what patterns exist in the data. So a very much discovery-oriented problem of I have some data, what's here? And the approaches we use to attack those questions are usually unsupervised algorithms. And then on the other side, we often have the questions around like what fits this pattern in the data. So I know I have group X and I have group Y, and I really want to know what separates group X from group Y. And we would approach these with something called supervised algorithms. And there's some other algorithms that kind of fit in at various points, but I think these are the two kind of predominant classes that are most often used right now. And so just to give you an idea of how these methods might work. So for unsupervised learning, we essentially have a whole bunch of unlabeled points. These should all be white on this canvas of, say, one axis and another axis that might be a couple features. And essentially what we're then trying to do is say, okay, what groups are here? And so, this hypothetical algorithm might say, actually, I think there's 3 groups here. There's the orange group, there's the green group, and then there's this blue group. And so that's kind of an unsupervised learning question. Like what are the fundamental patterns that are here? And just can you sort of group things and sort of return them to me? On the other hand, a supervised learning question will start with labels. So again, we have exactly the same points and we have exactly the same axes, but this time, instead of our points being all the same color and we're trying to discover the colors, here we start with green points and we start with orange points, and we're essentially trying to come up with a rule that will separate the green points from the orange points. And I think when we encounter these types of algorithms and approaches, it can be very tempting to say, oh, this problem is easy, so this other problem is easy. So this is an XKCD actually from about 6 years ago now. When a user takes a photo, the app should check whether they're in a national park. And the programmer says, sure, that's easy. GIS lookup, give me a few hours. And then the person says, and check whether the photo is a bird. And the difference between these two seems like essentially they're the same problem, but in reality, at the time, this was the difference between a GIS lookup, like basically a few hours of work and 5 years with a research team. And my hope is that by the end of this lecture, you'll at least have a little bit of intuition about whether what you're asking is a few hours of work question or a 5 years of research question. And I think often when we see what has been done elsewhere, we can sometimes think that we're looking at a few hour question, but many of these are actually going to turn into research questions. And hopefully you'll get some intuition as to why that is. The other thing that I know there's a fair amount of confusion on is what's the difference between machine learning and deep learning anyway? And you could certainly draw sort of bounds with certain classes of algorithms. I find that less helpful. What I like to think about is sort of, okay, intuitively, how are these approaches different? And the way I would usually define the difference is that for a machine learning algorithm, you have some input, and that input is something that's maybe not intrinsically meaningful to a human, but has values. So it could be pixels in a picture. It could be gene expression measurements in a sample. And then what you end up applying to that input is some sort of feature extraction method. And this might be, say, summarizing the input to pathways, if it's gene expression data. It might be, if it's an image, it might be running some sort of image detection question over it. So in the previous example, where we're trying to figure out if a photo is a bird, which is something I'm going to keep coming back to, you might have a rule that says, oh, if this texture exists, that texture, it looks a lot like feathers, so it would get a high feather score. These feature extraction algorithms then give you features, which you can then plug into a sort of standard machine learning method. There are many. I won't dive into what they are, but essentially they're all trying to build these decision rules using those input features. So for instance, featherness. And then from that, they're trying to predict an output. So give a probability of a bird. What differentiates a deep learning algorithm from a machine learning algorithm is often that, in this case, you've got your input and it's still this raw input. So like pixel level values from an image, you're essentially putting in the sort of how intense those pixels are. And then the deep learning method actually has to both take that input and create features with it, and then use those features to do a prediction task in a way that doesn't really have human intervention in here. So these feature extraction methods are generally human created and separate from the prediction method. Here it's all combined together. And this can be really powerful. It can give you the ability to solve challenges that we otherwise have thought are nearly impossible. But it also makes the predictions somewhat challenging to interpret and creates some kind of caveats downstream in their use. And so I think the power here is great, but there's also some caveats to be aware of, and I'll talk a lot more about these caveats in the future. And then you get this output step. So really what the deep learning method has done is it's essentially coupled taking the raw data and going to features with the prediction task. So it's essentially doing all of that at the same time. To do this, you need a lot more data, and but there's ways around that too that I'll talk about towards the end. So let's talk about check whether the photo is a bird, because I thought that was a nice example. So if we've got our machine learning algorithm, we're putting in a picture, and then we're going to apply some feature extraction methods on top of this. And we might imagine that we have a feature extraction method that's featherness, that essentially says, how feathery is this image? We might have another feature extraction method that's beakiness. It might say, how beaky is this image? And you might imagine that for many, many different tasks. And then what we're going to do is we're going to feed that into our machine learning algorithm. So we've done our feature extraction, and we now have a feather score, and we have a beak score. And we might say, okay, the orange points here were our photos with birds. So this one had a high beak score, but not a high feather score. These have kind of high beak and feather scores. And the green dots might be non-birds. And so what we'd essentially be asking is, can we sort of find something that separates these from these? And you might imagine intuitively how you would do this. Essentially, when you're picking a machine learning algorithm, what you're trying to pick is sort of how that decision rule should be constructed. If we imagine we have a new point here, which is this point in white, this question mark, we might say, okay, do I think this is a green point or an orange point? And this is the point where I would normally ask for input from the audience, but I know this isn't the right venue for that. But I might say, hey, I'm going to put the line here. This is my decision rule. It perfectly separated the solid green points from the solid orange points. Consequently, this is a green point. This is a non-bird. You might object to that. You might say, hey, I could have drawn a line here. It would be equivalent. And then this would have been an orange point. And really, the sort of various machine learning approaches are all essentially trying to learn how to build this decision surface. And different ones work in slightly different ways and have sort of slightly different costs associated with errors versus model simplicity. But essentially, this is the intuition behind it. You've essentially got some features, in this case, featherness and beakness, and you're trying to build a decision rule to separate the points that you know for these supervised algorithms to then classify new points like this one. And so, if this is our machine learning rule, we would say, okay, this machine learning algorithm says that this high feathery score, high beaky score point is really a bird. So that's kind of the traditional workflow. Deep learning, a little bit different. Really all of this stuff is combined together. Here's an example. This is a really nice example from Adit Deshpande that essentially takes a nice bird figure example and essentially shows how this is working. You've got these points here where you take a block of pixels and then you use those to create a feature. And you can do that through multiple levels of complexity. At the end, this is going to give you this set of features, which you're then going to use just like you would in the machine learning algorithm I showed before. You're essentially building that decision rule to create a prediction of whether or not something is, say, a bird. And the difference here is these things are, instead of in a machine learning algorithm, sort of traditional machine learning approach where these are all trained separately, in the deep learning approach, they're all trained together. So what's being shown here is this prediction task where sort of different parts of the image are being used and then integrated together to make this prediction. And that's sort of one way you use the neural network is after you've trained it, you can actually use that neural network to make new predictions. But when you're fitting it, you essentially have labels on bird. And so the extent to which something is correctly or incorrectly classified as bird actually, through a process called backpropagation, changes not just the sort of final weights at the end of the model that are sort of making where that decision rule falls, but they're actually changing the sort of fundamental structure of the features as well. So, it might be that you sort of have a feature that's vaguely feathery, but improving that feature might be a really good way to better predict bird. So that feature might become more and more optimized to sort of identifying that feather texture. So it's a way to use the full set of data to improve both your sort of feature construction portion and feature extraction portion and your prediction portion jointly. This clearly has some benefits. You can often get really nice performance with these approaches if you have enough data. It really makes kind of training data the key reagent to getting these things to work, along with some sort of model structure concerns. But it does mean that your models can become very difficult to interpret because the sort of features that it learns at this stage are not completely disconnected from sort of what happens at the second stage. So, where we might imagine we can sort of come up with the featherness figure, I'll show you later how we would love to intuitively have a featherness figure, but that can be somewhat hard to disentangle from sort of everything else that's going on in the neural network. And just a lot of the sort of advances in methodology here were driven through a reference data set called ImageNet. So this is ImageNet, which is essentially has photos of many different things. Here's a whole bunch that were in the kind of Viper category, and you might say, okay, these are not birds. But this data set has been has provided really remarkable examples of how deep learning algorithms can dramatically improve performance, in this case in sort of image analysis tasks. But over the last 5 to 10 years, over the last 5 years, we've seen this transform many, many different things from how we process and understand the written language. I think there's a lot of really nice examples in understanding genetic sequences and how they encode functions. So there's really, I think, a lot of that has been driven by kind of this initial starting point. So I wanted to note ImageNet here and just give you an example of how well these algorithms work and really where you see the sort of deep neural networks taking over. So ImageNet's been used in competitions for a while to sort of to understand how well an algorithm can say, find if a bird is there, in addition to sort of all the other categories that are in ImageNet. And traditionally, performance has been relatively static, except you can really see this rapid convergence over about a 5-year period here where deep neural networks really took off in this competition, with one of the first examples here of just sort of a new approach, this approach really kind of dominating the rest of the field in terms of performance, and how only a few years after that, these methods became as good as humans at sort of figuring out what object was in an image, which is really a remarkable improvement for a field that's actually quite old, it turns out, trying to understand what's in pictures. This has been utterly transformative. So, okay, we've been talking about birds a lot, and yet this is an Alex's Lemonade Stand Foundation talk. So why are we even thinking about birds? And it turns out these methods don't just work on birds, they don't just work on reptiles, they're being used often with images from healthcare as well. This is an article from a few years ago now that argues that a new algorithm can spot pneumonia better than a radiologist, which is a really provocative title. And we'll come back to this one in a little bit. This is a nice piece that really talks about, it sets up a contrast that I don't entirely love between artificial intelligence approaches and physicians, but it does ask a lot of very provocative questions, and has this quote from Jeff Hinton that I think who's one of the sort of pioneers of this specific set of techniques in machine learning that I think is at least provocative and should cause us to think. He argues this, that we should stop training radiologists right now. You know, everywhere you look, there's new examples of this. So just over a couple weeks last year, I ended up collecting a few different examples. This one argues that AI systems can ID ACL tears as well as radiologists. This one says, you know, can predict breast cancer up to 5 years in advance. This one essentially is supposed to be as good at human docs as spotting lung cancer developing. So really, you know, the argument is being made quite provocatively that these methods, which have revolutionized bird detection, are now going to revolutionize medicine. So, okay, this is the point where you might say, hey, I guess we're all done. You told me you were going to tell me about deep learning. You've told me a little bit about deep learning. You've given me a little bit of intuition of why it works. And you've now told me all these examples of how it's better than physicians at solving certain types of problems. So are we totally done here? Why are we still training radiologists, for instance? And I think it's important to note that while these algorithms have their strengths and they have certain areas that they excel at, they also have certain things that are challenging. So in this case, you know, I thought this was a really nice tweet. Determining if an image is a chihuahua or a blueberry muffin is a tough problem in artificial intelligence. You can really kind of see this here, right? The muffins have often a similar structure to the chihuahuas. In terms of the kind of dot patterning of blueberries on the muffin, and muffin doesn't look entirely dissimilar from a chihuahua face, at least to me. I'm sure there are people to whom they looked drastically different. But, you know, I think you could see why some of these would be a little bit difficult to predict. You know, if you think about them texturally, if you think about them from the point of view of colors, there are certain structural arrangements that make them distinct, but they're somewhat subtle. And you know, this type of image classification turns out to be actually reasonably difficult to get right. You can get it right in specific cases, but getting it right in the general case is actually not as easy as one would hope. There's some other things that there's some other properties that these methods have that are a little interesting. On the left here, this is a picture that I personally perceive to be a panda. So I perceive this to be a picture of a panda. The example neural network here also perceives that to be a panda with about 60% confidence. What we're going to do now is we're just going to add 7 1,000ths of this dot pattern to that image. So it's essentially, you know, this image with just this tiny little bit of dot pattern added. This dot pattern, you know, is not highly confidently predicted to be anything, but the highest confidence is a nematode with 8% confidence. So we run that. We create this image, which to me, I perceive to be no different than this. I perceive these two to be essentially equivalent images. However, if you are the neural network in question, it predicts this to be a gibbon with 99.3% confidence. So this is a little odd, right? If we were thinking about a human, you know, there's probably no radiologist that's going to look at this and say panda, and then look at this and say gibbon, right? So there's something else going on here. And what's going on here is this type of noise that's added is something called adversarial noise. And it's essentially looking for where that decision threshold is and really trying to push, it's making slight changes to those features, you know, making slight changes to the featherness feature, making slight changes to the birdness, to the beakness feature, to push something as over that decision threshold with really minor changes in the input space. So you can get a picture that looks effectively the same, but is called differently. And so I think this is important to think about, because while these methods may often be highly accurate, their predictions might actually be somewhat brittle. And maybe here it's just kind of funny that it's a panda and a gibbon, but if there are sort of systematic changes that, you know, perturb things in ways that are not terribly different than what one might be able to achieve with an adversarial approach, you could end up making the wrong decision with high confidence. And so it becomes really important to think about the extent to which your input space and training data really match your use case that you aim to apply things to. And this is just a fun one to talk about. So this is this paper put up on arXiv 3 years ago now, introduces these adversarial image patches, or you can actually have them as stickers. This is the toaster sticker. And then this is kind of fun. So I perceive this to be a picture of a banana on a table. The classifier, the neural network classifier, also gives it a high prediction score for banana, but maybe a secondary prediction for slug that's low confidence, and then not really much else. You can maybe see why that would be the prediction, particularly if it had been trained on banana slugs or something. If you think about it, if you then place this sort of sticker patch next to it, because it's the toaster sticker patch, it's really created to trigger that these neural networks will call a toaster as the output. And sure enough, when you do that and put this image in, you actually get a high toaster confidence score, and bananas really drop down. So I think this again kind of shows the brittleness of these methods, not just if you sort of add random patterns across the image, but you can actually just add if you add sort of patterns across the image, you can also achieve these kinds of bizarre predictions by adding sort of patterns to just a small part of an image. So the last one was sort of a small amount of change that was broadly localized across the image. This is a large amount of change, but localized to a small part of the image. And it's really in both of these cases that you see really substantial differences in kind of what the output is given relatively small changes in the input. Okay, so what's happening here? So yeah, let's talk about this, but let's think about a case where maybe it has a little bit more biomedical relevance. So this is that same piece that I started with, the algorithm that can spot pneumonia better than a radiologist. This is from a blog, like everything in machine learning. If you want to learn, if you want to see the latest work, this is a blog post from John Zek on Medium. What are radiological deep learning models actually learning? If you're interested in this, I would really recommend going to read that, because it's really, I think, a really insightful dive into the predictions from these methods. In this case, what he's done is he's taken a sample that has one of the highest probabilities in the data set of pneumonia. It's only a 5% probability from the classifier, but it's one of the highest in the data set. And he's asking, okay, why is this being predicted as pneumonia? What is actually driving this image to have that prediction? And where you see a positive score, that's increasing the sort of prediction, the pneumonia prediction. Where you see a negative score, that's not contributing to the prediction. It's actually reducing the likelihood that it would be called as pneumonia. And one of the things that you can see is really the most informative aspects of this image are the bottom here, which is sort of outside the lung cavity, and where you might not expect to be drawing most of your predictive strength from. And in fact, sort of things that are in the lung, especially regions of density in the lung, actually have sort of negative scores. So that's a little odd. And the hypothesis that's presented here is that it's, there's potentially a noise pattern that's scanner specific at the bottom of the image. And that noise pattern, may be associated with an X-ray that is more frequently used to examine patients who have pneumonia, just because of where it's potentially located in a hospital or the units that use it. And that may be what's driving some of these sort of predictions that are kind of a middling confidence, but among the higher end of all the predictions in the data set for pneumonia. So essentially our data set might actually be, some of the performance may be driven by biases that are sort of non-random distribution of which scanners are used with which phenotypes. And I think that points to some challenges of how we evaluate these models and how we really need to think about performance. Because you could imagine a physician is probably not going to spend most of their time looking at this part of the image to understand which scanner is being used and whether that increases or decreases the probability of pneumonia. What you probably want is something that's really focused on the salient parts of the image. Here's another one. This is there's a few of these in the blog, but I thought this one was interesting. Up here, this is also contributing to the probability of pneumonia, but it's just the word portable. So I think getting that that type of information where probably someone's shoulder and is not contributing to whether or not they have pneumonia. It's really this kind of keyword portable here that suggests that the person, the X-ray had to be brought to the person as opposed to the person being brought to the X-ray. That may be more suggestive. And these things all get confounded together. And so this is, a hazard that we really need to be aware of when we're applying these methods, because we really would want them to make predictions, exclusively from the lungs. And that's not what we're seeing in terms of performance. Like a human radiologist is likely to focus more on the lungs than, the word portable on an image. And that's probably a good thing. So there's a hashtag, and I think now a blog started by Janelle Shane. And what she observed was that frequently, if you use pictures of rolling green hills, regardless of whether or not there are sheep on them, neural networks will often call sheep. And consequently, if you put a sheep in an odd situation, even something that is kind of an obvious sheep. So here she asked for people to post pictures of sheep in odd situations. So here's a sheep drinking from a water fountain. The neural network returns for this one, a woman is holding a dog in a kitchen. And so you can see how maybe the sink would lead to that, and maybe the woman being here would lead to that prediction. But really, to many humans, I think we would say this is an obvious sheep, despite what it's doing. And again, I think this is highlighting the propensity of these methods to really learn from their background. So, to learn rolling green hills are often associated with sheep when humans take pictures. A certain X-ray is more often used with patients who have pneumonia. Those things all get baked into the model. And in reality, that's not what we want, but it is what's happening. And so we need to understand that if we're going to evaluate both the likelihood of success with these methods and also like what the performance really and truly means. And then there's another thing that I think is really important for us to think about, particularly as we start thinking about using these in medicine or in other kinds of decision-making areas. So this is an article that was a really kind of interesting premise. The argument that they made here was they were going to predict criminality simply from people's photo IDs. So they took their ID picture before any criminal conviction, then subsequently observed whether or not a person had had a criminal prediction, and they asked if they could predict a criminal prediction, if they could predict that someone would be a criminal solely based on their ID picture that was taken before any activity that would cause someone to be arrested. And so their argument is that these methods have no bias whatsoever because the training data are, because the pictures are unbiased and the neural network is unbiased. What these authors really did not consider, and it's critically important to consider, is that in fact what this does is it bakes every bias that we have in our society into these, because they're the biases of human judges, the biases of the police, the biases of every actor in the system is now baked into the training data. And so when we train these neural network models, we're essentially laundering that human bias into essentially bias within a computer model. And that's really, it lets us launder our bias, but it doesn't eliminate our bias. And so I think we really need to consider the extent to which our behavior in using these is going to create discriminatory outcomes and essentially perpetuate the injustices that we already have. And so I think it's really important to consider not that these have no biases, but in fact that these have all of our biases, and that we need to consciously work to mitigate those and to avoid having them baked into these models. And particularly as we're thinking about these being used in healthcare, we know there are different outcomes for individuals in our healthcare system by race. And if we really need to avoid using these machine learning models to essentially permanently build those into our system in a way that's very hard to get rid of. We should in fact try to use these types of approaches to avoid them. And really what a lot of this comes down to is this quote. So I was reading a blog post around sort of big data and p-values. And I got into the comments section and I saw this from someone who I know named Daniel Himmelstein, who used to be a postdoc in my lab and is now working at a startup. And he wrote, on big data, data collection biases are always larger than statistical uncertainty. And this is completely true. And it's really what's driving most of these caveats that I'm telling you about. Essentially, there are data collection biases. They are pervasive. If you're using a traditional sort of machine learning or statistical approach, you can really start asking questions around whether a prediction is being driven by things you should expect to drive the prediction, or whether it's being driven by something that might be associated with a bias. When you're using these deep neural networks, you really have to be highly, highly, highly proactive around how you examine them. And we often want to attribute sort of some understanding to a neural network. I've even done it throughout this talk at times where I'm like, oh, the neural network sees this, the neural network sees that. We have to be very careful about that. We can talk about it, but whenever we talk about it, we need to really understand that what we're saying is not precise. So this is an article from deep learning on Wikipedia. In an image recognition application, the raw input may be a matrix of pixels. The first representational layer may extract the pixels and encode edges. The second may compose and encode arrangements of edges. The third may encode a nose and eyes, and the fourth may recognize the image contains a face. This is a really attractive way to think about these methods. And it's very human intuitive to think about it this way. Unfortunately, it's also probably not right, and it's not right in ways that are a little bit confusing. So this is a really nice paper from Chris Olah and Distill and others. And so what they look at here is the pattern of a low-level neuron, and we could maybe imagine that this low-level neuron is essentially capturing edges, maybe with some spirals, although it looks like maybe there's some noses here. This is a single example. These are a few more examples that sort of really trigger this neuron, and then these are some examples in the data set. You can see why a neuron with this structure might, a neuron that finds these things might also find these things, right? You've got these curves here. You can see that kind of consistency between kind of chains and cranes, and it looks like snakes, or maybe necklaces. But then when you get to kind of higher level neurons, remember, these are the things that are supposed to be recognizing a face or a nose. This is what maximally activates these. And to me, this kind of looks like a cat and the hood of a car, and maybe certain types of dogs, right? And so that's really what you see, and that's what you see here. So you might think that, okay, this is sort of a textural thing, and it's going to sort of maybe recognize different types of image, but then you're going to get sort of a neuron that recognizes one thing and only one thing. And it's really not that. You can see how this second sort of higher level neuron is a composition of these, but it's really not exclusively recognizing one thing. And if we think about it that way, we're going to sort of think about things in a way that might lead us astray. And then the other nice example of this is a paper from ICLR last year. In this case, what they've done is they've taken images and they've essentially preserved the textures, but shuffled the structure. So there's an original image that I'm not yet showing you, and then they've texturized the images. It turns out that deep neural networks, which are theoretically learning structure at these higher level layers, like a face, are probably mostly learning textures. So doing this doesn't really reduce the prediction performance of these methods at all, even though you've destroyed the structure compared to the original image. And so I think it's really, really important to think about these things as they're going to learn the simplest features that distinguish the classes that you're asking them to distinguish, whether those are intrinsically meaningful to us or not. As much as we'd like to ascribe meaning to them, we can't take for granted that there's meaning there. If we do, we're going to be led astray. Okay, I've just told you all the problems with these. First, I sort of outlined the methods. I've told you a whole bunch of problems. So now the question is, okay, how do we actually do this in a way that's helpful? There's a few techniques that look like, to me, they're going to be particularly helpful in biology and medicine. So I'm going to talk about them. But first, here's the ideal world, right? This is the ImageNet world. You collect a whole bunch of stuff, millions of training examples. You just get as much stuff as you can, and then you label it really, really well. You put a bunch of human time into labeling. You get the best labeled examples you can possibly get. You feed these to your neural network. You feed these really clean data in, and you'll usually get really good prediction performance. So you'll be done. That's nice. Those of you who sort of work in biology and medicine know we don't live in that world. Even our true training examples are often not particularly clean, right? We don't have a single X-ray that gets used for every single person who comes into a single clinic. We don't have a single X-ray that gets used for every single person who comes into every hospital in the US. So we're just never going to get really that kind of level of cleanliness, that really nice level of kind of clean training data. So what do we actually do? This is the point where I'm going to list a few things that I see as generally useful. There's also many, 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 many field-specific techniques that rely on sort of having the combination of expertise around the problem, as well as the kind of combination of expertise in machine learning to drive the right approach. But this is essentially now where we hit the difference between the type of problem that's going to take a few hours and the type of problem that's a research problem. And in biology, most of the things we're going to deal with are still going to be research problems. So the first thing you can do is, in some cases, the problems that we work on have certain structure. In an image, the pixels of the image in one pixel often relates to its neighbors in a kind of deep and intrinsically meaningful way. Like in an image, something that's immediately neighboring a pixel is more likely to be relevant to that pixel than something that's far away. and we can encode that into a model. So we can use something called a convolutional neural network that uses this kind of local structure. And that can really, really, really help when we are building a model, as opposed to something called, this is on the left, a multi-layer perceptron, where everything is fully connected and it sort of imposes no structure. This model is much more flexible. However, if the problem meets the convolutional assumptions and we use a convolutional neural network, we're likely to get very good performance with many fewer examples. This one may require more examples than we can possibly generate. And so really thinking about the structure of the problem and the structure of neural networks is really helpful. So this is like thinking about the structure of your solution. The next technique that's really worth thinking about is something called data augmentation. This technique is, instead of thinking about the structure of your problem, you're thinking about the structure of your training data. So if, for instance, I wanted to develop a classifier that could predict if someone is kicking a ball, I might have this picture with a foot coming towards a ball, and I might say, okay, that's one of my examples for a foot kicking a ball. But then what I can do is I can take that picture and I can essentially rotate it arbitrarily, because it doesn't matter if the foot is upside down, it doesn't matter if the foot is sideways. Essentially, as long as the foot is kicking a ball, it's relevant. So I can take my limited number of training examples, think about the structure of the problem, and then modify my training examples in a way that preserves the salient structure of the problem, the foot and the ball, but eliminates the less important parts of the less important structure of the training data, like the orientation of the foot. And in fact, this picture that I'm showing you, the foot is in fact not positioned like this. This is actually the picture, right? So the person's suspended in midair. So this is one of the reasons you really want to do this rotation, and think about how you can do this type of thing. And data augmentation is most commonly used with image data. My lab has developed and sort of written a short report. It's just on GitHub. We haven't published it, around how you can use this kind of data augmentation with RNA-seq data by using sort of sampling of reads. There's a whole bunch of different techniques, but really I would say fundamentally it's important to think about the structure of your data and ask if there are ways that you can change your data that preserve the relevant portions of the structure and completely alter the irrelevant portions. So this requires both a little bit of understanding of machine learning, but also a deep understanding of the problem. This is one of the things that's sometimes counterintuitive to people when they start to work in this field. It turns out that sometimes doing one thing at a time can be a lot harder than doing many things at a time. Another way to say this is that neural networks might be the ultimate multitaskers as long as those tasks are somewhat related to each other. And if you think about this intuitively, you can kind of understand why this is. Maybe here with one task, what we're trying to do is predict patient outcome on drug class A. And what we're trying to do here is predict patient outcome on drug class B. Well, maybe if we could take data and we could summarize it into a level of sort of pathway activity, that summarization into pathway activity might be similar across both of these tasks, and it's really only the final prediction step that's different. So if you tried to predict outcomes with group 1 and group 2, you get a lot more training data, and the common parts of the problem can be used to fit these early layers of this model. So layers 1, 2, and 3 can really share structure and benefit from multiple training examples. So this lets you get a lot more value out of a limited number of samples, which is the case that we often experience. And I know Jay introduced that Jacqueline Tironi, so Dr. Tironi is going to talk on Thursday about using transfer learning and how that can apply in the rare disease space. This is this is an example of, essentially having a neural network that's designed to solve one task. Often these low-level features can be reused in really important ways. So you can take those features, move them over to an entirely different task or an entirely different data set and still get a lot of value out of them. And in fact, have those predictions coming out of that be better than if you had just trained them to solve this task. Because often you may have too little training data for a second task, but you may have a lot of training data for a first task. And so this is essentially a way to reuse your data and reuse your machine learning models to get a lot more value than you could get with just trying to solve your current task alone. And then you can also do the second step of this, which is you can actually then use just the task-specific data to go back and refine your model as well. I've given you this sort of overview of what I think are kind of the really big ways that we can really quickly think about how to solve problems in this space. One is thinking about our problem and the structure of that problem and how the neural network can match that. Two is thinking about our data and the features of the data that we do and don't want to preserve, and using the features that we don't want to preserve to design sort of data augmentation strategies. And the third way is thinking about related problems and then using those related problems to help bootstrap ourselves to solutions for the problems that we really care about, using either multitask or transfer learning. And so if you're interested in this, we wrote a paper a few years ago that's a little bit of a tome. It's on the order of 500-ish citations, 500-ish references, and 40 or so pages in the printed journal form. But this is available if you really want to deep dive into many of the techniques that I talked about. But yeah, and then the final question that I want to just briefly raise is, so this is all sort of how to get these things to work in a research setting, but ultimately we want them to work in the real world. And this means, I've already told you you have to think deeply about the problem to get them to work, but you also have to think really deeply about the problem if you want to understand how they're going to work in the real world. And that's because of the way these methods are trained, the features that are extracted are really tightly coupled to the prediction performance. And if your training data is, say, cleaner than real world data, because your training data is collected from a certain set of hospitals that have sort of world renown with dealing with a certain type of condition, that may be different than the data that's going to come in from, a community clinic. And that's fine, but if you don't take that into account, your machine learning model is going to not deliver predictions that are going to generalize to the real world. The other question is, do the training data fully cover real world inputs? So, we might build a machine learning model to predict X from, the type of inputs that are seen in a certain type of clinic. If this model then gets deployed to sort of other places that might see that condition, but that, don't routinely see that condition, inputs might be fed in that are different than what the model is created to cover, and that generalization space may not be big enough. It's really important to think about what your biggest confounders are and to implement controls for these. So in the radiograph example where the bottom of the radiograph was driving at least some of the prediction, one way to evaluate this is really going to be to try to preserve the sort of within unit structure of these, but to try to eliminate the confounders there, like the sort of particular machine that's used either by subsampling or oversampling, or by trying to collect an independent test data sets that are unlikely to have the same confounders. And then this takes us to our final point, which is that whatever you do, you cannot leak test data into your training set. If you leak test data into your training set, you will overestimate performance. We often think about this as happening when we just include the same, like maybe you have two X-rays of the same person, but they were two different radiographs, so you included them separately, and one of them ended up in your training set, one ended up in your test set. That's a big problem, and you need to be aware of that, and you really need to avoid that. But this can also happen in more subtle ways, like, okay, I built a machine learning model and its performance wasn't that good. So I went back and I used data augmentation. And when I went back and when I said it wasn't that good, I mean, I tried it on my test set, it wasn't that good. So then I went back and I used data augmentation, it got better. And then I changed my neural network structure and it got better. And when I'm saying it got better, I'm using my test set for evaluation. In this case, I'm actually closing the loop on the test set and incorrectly using the test set multiple times. And if you do that, you're going to overestimate your performance in ways that are really, really dangerous. And so making sure that whatever your test set is, you only touch it once is really important in these settings. There are techniques to address this in sort of more sophisticated ways. In the interest of time, and given how sort of technically involved those techniques are, I didn't put them in here. So I'm just leaving it at the advice of really, really, really use your test set only once. And then finally, I just thought this was interesting, so I wanted to put it in just as some food for thought. I've been talking about how these neural networks make predictions in ways that are very different than us. This is a paper that came out a couple years ago, and I had the opportunity to see it presented at a meeting, I guess right as it came out. And it just kind of blew my mind. So this is a picture. I perceive this picture to be a German Shepherd, but in fact what this picture is it's a picture of something else that has had noise added to it, this adversarial noise, like the gibbon panda example, to make a neural network make an incorrect classification. And in fact, in this case, it was this picture of this Siamese cat, and this noise that was added, it does fool a neural network, but it also fools for many humans. It fools us, and it gives us this long-term perception that this is in fact a German Shepherd. I know for sometimes when I present this, a subset of people are like, that's definitely a Siamese cat, and another subset of people always see it as a German Shepherd. I'm in the German Shepherd crowd. So whatever's happening in this image and however this image is being perturbed, I just think it's really interesting that these neural networks often make predictions in very different ways than humans do, but every once in a while, the things that can drive a picture to an interesting classification in a neural network can also do that in our human neural network and sort of our visual system as well. So I thought that was interesting. I want to leave some time for questions, especially since I have a hard stop at 2. So I just want to thank the people who I get to work with at the Childhood Cancer Data Lab, the data science team. So Jacqueline Taroni is going to talk to you on Thursday if you come in and listen about transfer learning and how that can be applied in the rare disease and pediatric cancer space, now that you've kind of gotten the primer of what these approaches are and how they might work. So I just want to highlight that as an upcoming talk, but really this whole team of people that I get to work with is just wonderful. And hopefully you'll look into this Childhood Cancer Data Lab and what we do, and I'd be happy to answer the questions that folks have.
All right, Casey, that was awesome. Thank you very much. If I'm hearing you correctly, we're going to work on a bird song and bird photo identifying thing on the side.
Yeah, I did have to put the bird in, given your previous questions around identifying bird songs.
Yeah. Okay, so the first question comes in from Shayla, and she says, can you please explain what exactly a neural network is?
Yeah. So I glossed over that here because the answer is deeply unsatisfying in many ways. So a neural network is essentially a system of mathematical transformations that are applied. And they have many different structures, but the ones that I've noted here are something called feed-forward neural networks. The idea is that these were designed to originally work similarly to how neurons worked, in that you have some input, you have some synapses, those synapses can be strengthened or weakened, and there's some propagation through that. But I find that that analogy holds up less and less well as a field of neuroscience advances. So I guess I would describe them as 2 ways, either essentially a set of mathematical functions that build on the output of the previous function, the previous functions, or kind of the best analogy would be akin to how a computer scientist would have thought neuroscience worked 50 years ago. I guess 70 years ago.
All right, well, Case, we got a lot of questions here, so we're going to try and get through as many as we can in the next 9 minutes. Robert Alloway asks, what are your thoughts on assembling different methods with distinct biases? Does this dilute the effect of individual biases, or is the result just a model with all of the biases?
That's a good question. In the interest of answering this question quickly, I think what I would say is, if we have a really strong prior belief about what the structure of a correct solution is, given our understanding of the problem, we should use that structure. If we do not have a strong belief about what that structure should look like, I think it would be better to use an ensemble of many different models or many different approaches to try to get some sort of consensus. The ultra short answer to the complex question.
Okay, Arthur Frank asks, does rotation and other augmentation methods eventually lead to overfitting, or does the CNN truly look at the image differently?
It should reduce the impact of overfitting for a while. You run the risk of, you run this risk even if you don't do it, of potentially memorizing your input data. What really helps is just having, if your signal just has to be distinguishable from the sort of individual noise of the training data. And augmentation will help because, it removes some of the sort of directional noise. So like any of those translations and transformations you can do help to reduce the sort of systematic noise and hopefully highlight the signal, but it's not going to completely eliminate it. It should just help a little bit.
Okay, next question comes from Steve Lesnick, one of our scientific advisory board members. The brittleness of a decision reminds me about issues for overtraining models, particularly around gene expression data. It seems to me that deep learning at its core is highly dependent on overtraining. Example, recursive learning processes. How does that translate when you move from good training data to noisy real life data? Is that why the output breaks?
Yeah, so The specific examples that I showed were adding certain types of noise that were especially created to induce brittleness. So those levels of brittleness were certainly not representative of what you'd see just from kind of moving from one noise source to another. I wouldn't probably think about these as necessarily being inherently more brittle at the decision phase than I might think about another machine learning method being. But what I would say is that because the features are kind of tightly connected to that input phase, there is potentially a little bit more brittleness if there's like a substantial domain shift, especially if the sort of features that are used in the feature extraction stage are generally gathered from many data sets. So I think the best way around this is to include as many different cohorts that have systematic noise as possible when you're trying to do this training, to really try to break the connection between some systematic noise source and predicted output.
Okay, Case. The next question comes from Arthur Frank again. When dealing with pediatric cancer in the small number of samples, how do you deal with the lack of data size?
Yeah, so tune in to Jackie's talk on Thursday, and she will give you a set of solutions that may be helpful in many cases. You can see from her title that she's using one of the transfer learning techniques that I mentioned.
Okay. Next question comes from Charlampus Lazarus. Thank you very much, Casey, for this nice introduction. I'm wondering, based on your experience, if you could share some examples of problems in the field of childhood cancer epigenetics, transcriptomics you have solved using deep learning, and you would be able to solve, and you would be unable to solve otherwise.
Yeah, so I think we're at the pretty early days there, and as you note, the sample size is lower in those cases, even than many of the examples that I've talked about here. I think the imaging world, if you're thinking about imaging, that's going to be one of the areas where advances are likely to happen sooner, just because of how much strong structure there is in images and how much can be shared with sort of other image modalities. And then again, I'll advertise Dr. Taroni's talk on Thursday as they're not deep learning methods, and that's kind of so that's by intent, but how you might think about using these transfer learning approaches to sort of start to address those challenges. And Jackie may be able to also discuss why she didn't use a deep learning approach at the end of the day for that problem.
Okay, the next question comes in from Rich Abdil. It says, hi, Dr. Greene, thanks for an interesting talk. You mentioned convolutional neural networks. I'm curious about your thoughts on how they might be used in the context of biological data. If the idea of proximity in an image is the distance between two pixels, could the same approach be taken in, say, a microbiome data set by using phylogenetic relationships, or a gene expression data set by using pathways?
Let's see. Okay, interesting question. So I think the key is, if you're going to use a convolutional approach, meeting the convolutional assumption. There's a series of papers now that have come out. I think one of the earliest ones is from Jan LeCun's group, maybe 4 or 5 years ago, about using convolution over graphs. So the convolutional approaches I talked about were pixels in an image, right? So kind of the surrounding cells of an image. But there's also techniques now for working on convolution over graphs. So if you can define a graph and you know that sort of the information about a node in the graph is more similar to its neighbors in that graph than sort of other parts of the graph, I think that's going to be how many of these problems that are not image-based are going to be more addressable with convolution. But I would just point you towards that as an area of literature in the interest of a quick answer.
All right, Case, I don't understand this next question, but I'm going to read it to you. Hendrik Lim says, some people said that machine learning is a black box. Is there any way recently to make it at least not black again?
Yeah, so I think there's a whole bunch of methods that people are working on to improve sort of saliency. So how we understand what predictions led to a certain outcome in a specific case. I guess I would just say fundamentally, do not forget the principles of experimental design. Essentially, I think being able to figure out ideally kind of where a prediction is coming from, make some predictable perturbation to that, and then observe some predictable change in output is a helpful way to kind of understand what's happening here. And there are a whole bunch of saliency methods that are kind of based on that. So I guess I would say we're getting a lot better at sort of understanding which individual features are leading to predictions in certain cases. But I think ultimately where I see these being used, especially in kind of the basic discovery place, is it's sort of helping to integrate a large amount of data and then plan the next experiment. But having the findings come back to the lab, like the idea, really bringing the finding back to sort of the basic research lab, if you're thinking about using it in that setting, and then making predictions from the model and testing them, I think is one of the kind of only ways to approach that. Hopefully that's helpful.
Okay, I guess we have time for one more case. Can you explain some examples of scientific problems that would benefit more from a machine learning approach versus a deep learning and vice versa?
Yeah, so I think the pediatric cancer example, especially with genomic data, is great, right? For gene expression data at the moment, we don't have a lot of understanding of what a convolutional operator would be. And for, if we're trying to say predict what drug someone would respond to, or if we're trying to predict the outcome of a clinical trial, right, there's a really small number of samples. And so these are cases where, like Dr. Tironi will talk about on Thursday, these kinds of linear or models that have sort of a non-linearity, but that's kind of simple, can be really, really advantageous. I think we are moving more and more towards the world, especially with image data, where we are starting to be able to compile enough data from different problems to start repurposing it for the problems we really care about. But that's going to be further away. So I think the earlier gains are going to be using these same types of techniques, transfer learning and whatnot, with more traditional sort of machine learning approaches, and then understanding what works there and then leveraging that into the deep learning space.
All right, Case, we had a lot more questions. I know you have to go. For those of you whose questions did not get answered, maybe we'll figure out a way for Casey to answer some of them. Maybe we'll post it on one of our Twitter accounts or something.
Yeah, or a blog post or something.
Yeah, or a blog post. That would be good on the refine.bio or CCDL blog. Thank you, everybody, for joining us. Thank you, Casey, for the amazing talk. And please tune in on Thursday for Dr. Jacqueline Tironi's talk on transfer learning.
Thank you so much.
All right, thanks, Case.